డాక్టర్ బిఆర్ అంబేద్కర్ సార్వత్రిక విశ్వవిద్యాలయం విద్యార్థులకు స్వాగతం. ఈ ప్రసారంలో
Welcome to Dr. B.R. Ambedkar Open University UG Botany classes. Today's topic is structure and reproduction of bacteria. When you are going to look at bacteria, bacteria is a unicellular prokaryon. Why we are calling it as a prokaryon means prokaryon is a primitive cell. We have another cell called as eukaryote. There is a little bit difference between eukaryote and prokaryote. Karyon means nucleus. The cell which has a definite nucleus, that is called as eukaryote. The cell which does not have a definite nucleus, that is called as prokaryon. One of the best example of prokaryon is bacteria. When you are going to look at bacteria, bacteria is a omnipresent. When you are going to look here, bacteria is going to present in water, soil, air, food, skin, inside the body, on most of the objects on the earth. And also the shape of the bacterium is governed by its rigid cell wall. However, exactly what attribute of this rigid cell material determines that a cell have a particular shape, and it is not yet understood, but the bacterial cell are with different shapes. So, when you are going to see, there are different types of bacterial shapes based on their cell wall. So, when you look here, there are three different bacterial shapes we can observe here. First one is spirilla, next one is rod shaped, next one is round shaped. Spiral is example for spirilla, and rod shaped we are also called as bacillus or bacilli, and round shaped we are going to call as cocci. So, when you are going to observe here, there are three different examples of bacterial shapes. Bacillus anthracis is a best example of bacillus, Neisseria meningitis is a best example of cocci, and Leptospora is a best example of spirulina. Major structures of bacterial cell, if you are going to discuss about the structure of bacterial cell, structure of bacterial cell is divided into three types. So, that is external to the cell wall, internal to the cell wall, and cell wall. When you are going to observe cell wall, the external cell wall consists of few things. Those are flagella, capsule, and pili. These three are the external to the cell wall. So, when you are going to observe the ultrastructure of bacteria, these three are included as external structures of bacteria. So, when you are going to look at flagella, flagella is an external structure. It is a whip like tail that some bacteria have for locomotion. So, it is a hair like and helical appendages that protrude through the cell wall and are responsible for swimming and motility. They are in 0.1 to 0.2 micrometers in diameter. It is composed of three parts. One is basal body, next one is hook, another one is filament. So, mainly the flagella is made up of a protein material called as capsule, and it is mainly used for motility of the bacteria, and it also used for the movement of the bacteria from one place to another place. These movements based on their responses may be chemotaxis, phototaxis, and magnetotaxis. When you are going to look at next one, that is capsule. Capsule is a outer layer. It is a viscous material forming a cover layer around the cell wall. It appears to be amorphous and gelatinous area surrounding a cell. They helps in bacteria against temporary drying and desiccation and it blocks the attachment of bacteriophages to the cell surface. So, what we can usually say about capsule is, capsule provides escape mechanism for bacteria from the virus. Next one is pili. Pili is one of the external structure. It is hollow, non-helical, filamentous appendages that are thinner, shorter and more numerous than flagella. They do not function in motility, but associated with bacterial mating and attaches to the epithelial cells. These hair-like materials mainly helps in attaches to the surfaces of the epithelial cells and for mating of bacteria with other bacteria. So, these are some outer to the bacterial cell, these are flagella, capsule and pili. And when you are going to discuss about cell wall is a very typical for bacterial cell, because the structure and rigidity of a particular bacterial cell depends upon the composition of the cell wall. The cell wall of bacterial cell is categorized into two types based on a invention of the staining technique by a Christian Grams. So, the bacterial cell wall categorized into two types, that is gram positive and gram negative. If you see here, the outer covering that maintains the overall shape of the bacterial cell, that is cell wall. So, when you are going to look at functions of the cell wall, it provides shape and it protects the bacteria from osmotic shock and it contributes to the pathogenicity and also it protects from toxic compounds. So, as I said earlier, the bacterial cell wall is differentiated into two types, gram positive and gram negative. Let us see gram positive. Gram positive bacteria has a cell wall containing relatively large amount of peptidoglycan. So, peptidoglycan is a material mainly made up of sugars and amino acids. The main sugars are heteropolysaccharides. Those are N-acetyl-glucosamine, N-acetyl-muramic acid, and they are linked by L-alanine, D-alanine and diaminopimelic acid and lactic acid. So, when you are going to observe here, this is the bacterial gram positive cell wall. All these are peptidoglycan units. Often these contains teichoic acids. These are linked to muramic acid of peptidoglycan with phosphodiester bonds. When you are going to look at cell wall of gram positive bacteria, these are teichoic acids and these are peptidoglycan units. They will provide a rigidity and structure to the bacterial cell wall. And then when it comes to gram negative bacteria are more complex chemically than gram negative cell walls. It mainly consists of lipopolysaccharides. Lipopolysaccharides is a material mainly present in the gram negative bacteria. This lipopolysaccharides consist of lipid A, core polysaccharide and O antigens. These are the three units combined together called as lipopolysaccharides. Lipopolysaccharide content is more than 80% in gram negative cell wall. So, the main functions of lipopolysaccharide is they stabilizes the osmotic mobility and also the O side chain composition can be changed to avoid the host antibody detection. Means the O antigen helps the bacteria to escape from the antibody activity of a host system and also it provide negative changes on the cell. So, when you are going to look at here, this is the cell wall of gram negative bacteria. It consists of lipopolysaccharides and this is the peptidoglycan content. It consists of around 20% of the peptidoglycan content. So, by this what we can say about cell wall of the bacteria is that cell wall of bacteria can be differentiated into gram positive and gram negative based on the staining technique called as gram staining. This is the procedure of gram staining technique by which we can differentiate the cell wall and by this we can know the composition of the cell wall. So far we have discussed about ultrastructural bacteria. In that we discussed about external to the cell wall and cell wall. Now it is internal to the cell wall. Internal to the cell wall we have cell membrane, cytoplasm and nucleus. Now we will discuss about cell membrane. Cell membrane also called as plasma membrane. It is a semi-permeable membrane, consists of bilipid membrane where protein material is sandwiched by lipid material. Cell membrane is non-polar in nature and it is embedded with two kinds of proteins, peripheral proteins and integral proteins. When you are going to look at the bacterial membrane is a key for bacteria because most of the activities will take place in the membrane. For example, bacterial photosynthesis, bacterial transportation of nutrients and most of the activities takes place through the cell membrane. So, cell membrane is a key part for bacteria to sustain its life, and beneath the membrane we have cytoplasm. Cytoplasm, usually when you are going to observe the cytoplasm of the bacteria and cytoplasm of the eukaryotes, the cytoplasm of eukaryotes consists of many cytoplasmic organelles, whereas the cytoplasm of prokaryote consists of very few organelles, because bacteria is itself called as a primitive cell. Primitive cell is not that much developed as eukaryotic cell. That is why it consists of very few cell organelles. In that we have ribosomes. Ribosomes are very small substances present in the cytoplasm. They helps in the protein synthesis of the bacteria. The ribosomes are of 70S type. 70S type means their sedimentation coefficient is of 70. When you are going to look at the eukaryotic ribosomes are 80S. So, there is a difference between 70S and 80S. These 70S ribosomes are going to present in the prokaryotes, that is bacteria, and 80S ribosomes are going to present in eukaryotes, so called plant cells and animal cells. In cytoplasm we have storage granules, mainly helps in storage of the food materials. These are called as metachromatic granules and glycogen granules. These granules are going to present in the bacteria for storage of the food material. And apart from this, in the cytoplasm of the bacteria we have plasmids. Plasmids are called as small circular double stranded self replicating DNA molecules. These are mainly present in the cytoplasm of the bacteria, and this helps the bacteria for their extra activities, like the plasmids which are present in this cytoplasm of the bacteria can be categorized as F plasmids, R plasmids and col plasmids. F plasmids are mainly helps for the bacteria to mating with another bacteria, and R plasmids helps the bacteria for gaining the resistance from the antibiotics, and col plasmids helps the bacteria to elicit some of the toxins from it. So, these are the few cytoplasmic organelles which are present in the cytoplasm of the bacteria. And then when you are going to look at here, this is a cytoplasm. It is a jelly like material that make up most of the cell, and these are the ribosomes which are going to present in the bacterial cells. And next one is nucleoid. So, when you are going to discuss internal to the cell wall, next one is nucleoid. Why I am saying nucleoid? Usually in eukaryotes we are going to call it as nucleus. Why it is called as nucleus? Why it is called as nucleoid? In highly developed cell like eukaryote, the nucleus we are going to call it as nucleus because there is a clear membrane around the nucleus which will clearly separates from cytoplasm to the nucleus. Whereas in prokaryotes we will not observe any nuclear membrane around the nucleus which it will separates from cytoplasm of the bacterial cell. That is why there is no proper nuclear membrane around the bacterial nucleus, and it is integrated in the cytoplasm. That is why it is going to call it as a nucleoid. Nucleoid mainly consists of nuclear material, that is DNA. DNA is nothing but deoxyribonucleic acid, which will act as a hereditary material to the bacterial cell. Mainly the DNA present in the nucleus is in circular form. This is about structure of the bacteria, and now we will discuss about reproduction of the bacteria. So, when you see here, bacteria can reproduce in one of the two ways. One is asexual reproduction, another one is sexual reproduction. So, when you are going to look at here, asexual reproduction can be categorized into three types: binary fission, budding and fragmentation. In binary fission, one parent involved, offsprings are identical to the parent and each other. The advantages of asexual reproduction is simple, only one parent, and offsprings are fully formed and it is very fast. So, here a single cell divides after developing a transverse septum. This is called as asexual reproduction, that is binary fission. And next one is budding. Budding is a process by which a small bud is going to protrude from the cell, and this bud is going to separate it from the cell, and it can be called as a daughter cell, that is called as budding. Next one is fragmentation. A few of the organisms will enlarge, means most of the filamentous organisms will enlarge, so that in middle of it fragmentation comes, and where the fragmented cell has fragmented, and it will gives rise to new cell. So, these are the three main techniques: binary fission, fragmentation, budding will comes under asexual reproduction. So, these are some photographs of binary fission, how both the cell combines together and they are going to generate daughter cells. And next one is sexual reproduction. It is apparent that mating or conjugation in bacteria is radically different from sexual mating in higher organisms. It is not a reproductive process that occurs regularly at each generation. It does not involve meiosis, since bacterial cells are haploid, nor does it involve the fusion of gametes. Instead, it involves the transfer of some DNA from one cell to another, followed by separation of the mating of cells. While conjugation happens, the entire chromosome will be transferred from one cell to the another cell. So, here as I discussed in the structure of the bacteria, there are circular material is going to present in the cytoplasm of the bacteria, those are called as plasmids. In that there is a F plasmid which is responsible for the forming of the conjugation tube with another bacterial cell. So, here bacterial conjugation can be done by the bacteria which have F plasmids in it. So, this F plasmids responsible for the formation of conjugation tube with the other cell. The bacteria which have F plasmids in it, that is called as F plus cell or male cell. The bacteria which does not have the F plasmid, those are called as female or recipient cells. These male and female cells will come closer and they will bind each other with a tube called as conjugation tube. After the formation of the conjugation tube, the genetic material will transfer from one bacterial cell to the other bacterial cell. So, this is the way how the genetic material will transfer from one bacterial cell to the other bacterial cell. So, apart from the conjugation, there are two other mechanisms of gene transfer, those are transformation and transduction. Transformation is nothing but a naked DNA entering into the bacteria in its environment is called as transformation, and transduction means a transfer of DNA material by the association of virus, those are called as transduction. So, all these things can be included in the gene transfer mechanisms, and also we can call them as reproductive process of bacteria. So, when you are going to conclude the lesson, the bacteria is a omnipresent. It consists of various structures based on their cell wall. So, the bacterial cell shape will be determined by the composition of the cell wall. So, when you are going to summarize this topic, the bacterial cell is of different shapes with cocci, bacilli and spirilla, and when you are going to see the ultrastructure of the bacteria, and we can discuss external to the cell wall. In external to the cell wall we have flagella, villi and capsule, and if you are going to discuss about the cell wall is of two types based on Gram staining technique. Those are gram positive and gram negative. Gram positive of peptidoglycan content of 80% and gram negative is lipopolysaccharide content is of 80%. And when comes to the inside of the means internal to the cell wall, we have the plasma membrane. Plasma membrane is used for vital activities of the bacterial cell, and beneath the plasma membrane we have cytoplasm. Cytoplasm consists of cytoplasmic organelles like ribosomes, mesosomes and storage granules, and some of the extra chromosomal DNA materials called as plasmids. After the cytoplasm we have nucleus, and nucleus is in the integrated form in the cytoplasm, that is called as nucleoid. And if you are going to discuss about the reproduction of the bacteria usually takes place in two ways. One is sexual and asexual reproduction. In asexual reproduction we have budding, fragmentation and binary fission. In sexual reproduction we have conjugation and other gene transfer mechanisms, transformation and transduction.
మా కార్యక్రమాలపై మీ సూచనలు సలహాలు ఆహ్వానిస్తున్నాం. మీ సూచనలు సలహాలు మాకు తెలియజేయవలసిన చిరునామా: డైరెక్టర్, ఆడియో విజువల్ ప్రొడక్షన్ అండ్ రీసెర్చ్ సెంటర్, డాక్టర్ బిఆర్ అంబేద్కర్ ఓపెన్ యూనివర్సిటీ, ప్రొఫెసర్ జి రామరెడ్డి మార్గ్, రోడ్ నంబర్ 46, జూబ్లీ హిల్స్, హైదరాబాద్ 500033.